but not today. Today's gonna be great. Okay, I'm recording now and I'm gonna share my screen. Cool. Move it all to the side. All right. Okay, so the first topic that's on the exam is shade avoidance. And for shade avoidance, you need to know the two flow diagrams of how plants don't elongate when there's lots of daylight versus how they do elongate their stems in the shade. And then the third thing you have to know is the trade-off between elongation and defense that happens in the shade. So we can go through those flow diagrams. So the first one, we're gonna look at, oops, this is my eraser, is daylight. So in the daylight, what we're gonna get is no elongation. Ooh. No elongation. So the flow diagram that you should know is in daylight, there's going to be a high level of red light as opposed to far red light. So you could write that as a high red to far red ratio. When you have more red light than far red light, that's going to cause phyte to switch to the PFR form. So phytochrome is a photoreceptor that switches between the PR and PFR form. If there's a lot of red light, like during the day, it changes to the PFR form. If there's a lot of shade, there's more far red light and it turns to the PR form. So in daylight, it's in the PFR form. And then when it turns to the PFR form, you just need to know that causes a signal cascade. And in response to that signal cascade, pin proteins move to the bottom of shoot cells. So pins move to bottom. So if this is like a plant cell in the shoot, the little pin protein is going to move down here. And that's important because pin proteins are the ones that efflux auxin out. So auxin gets made at the top of plants, and then it travels down the shoot. And when it goes into a shoot cell, it's going to leave out wherever the pin protein says to go out. So if the pin proteins are here at the bottom, auxin is just going to travel right through the shoot. So because of that, auxin doesn't build up in the shoot. So no auxin accumulation. In the shoot. And that's pretty much it. So if there's daylight around, we're not going to get any auxin accumulation in the shoot. And then you could end it by saying no elongation. Oops. In the shoot. Okay, so then what would the response be in the case of shade? So in the shade, we would expect, yes, there should be elongation occurring. Oops. So in the shade, we decrease the red to far red light ratio. There's more far red than there is red light. So because of that, phytochrome is going to be converted to the PR form. And just like before, that's going to trigger a signal cascade. But now it's a different signal cascade. So the signal cascade that gets activated results in pin proteins moving to the sides. So pin proteins are now on the sides. And if you have a big stem with a bunch of shoot cells and all of their pin proteins are on the side, but not the bottoms, then when auxin comes in, auxin's going to tend to accumulate in the shoots. So we get auxin accumulation. In shoots. So what happens then? So auxin is going to bind a receptor in the nucleus of the shoot cells. Oops, binds receptor in nucleus. And then auxin plus its receptor is going to kick off the repressor of the Sauer gene. Kicks off Sauer repressor. Oops. So now we have disinhibition of the Sauer gene. We can transcribe Sauer, and Sauer proteins act by inhibiting phosphatase. And phosphatase basically inhibits the proton pumps by removing a phosphate. So if the phosphatase isn't working, it's not actively turning off the hydrogen pumps, and you get an increase in the hydrogen pump activity. Yes, question.
Would we have to include the Sauer phosphatase step, or can it be like implied just by saying that auxin and the receptor kick off Sauer repressor?
I would include what Sauer does.
Okay, thank you.
You're welcome. And so then when you get the increased hydrogen pump activity, it's basically just the acid growth hypothesis from here for why we actually get shoot cells elongating. So one thing increasing the hydrogen pump activity does is it creates a increased electrochemical gradient or hydrogen across the cell. Because we're pumping a really high amount of hydrogen out, there's less hydrogen in. Now we have a gradient. That's important because we can take advantage of that proton gradient and use it for active transport of chloride. So the hydrogen chloride symporter takes advantage of that gradient and causes chloride ions to go in. The other thing the increased hydrogen pump activity does is that hyperpolarizes the cell. cell. And what that does is you can kind of imagine in your head, and I've just been telling people this number, maybe it gets it to around negative 130 or 135 millivolts. And that is hyperpolarized enough to activate hyperpolarization gated potassium channels. Hyperpolarization gated potassium channels. Oops, my flux is on. Let me turn that off. And then in normal physiological conditions, the equilibrium potential for potassium is maybe around like negative 80 millivolts. So if the membrane potential is negative 35, that's going to cause a large electrical chemical gradient for potassium to move inwards. So potassium is going to move in because potassium wants the membrane at negative 80, and instead it's very much below that. So the result of both of these ion moving in is we get an increased solute concentration. And that causes a decreased solute potential. Well, what cells again? These are the shoot cells or the stem cells. I feel like you should say shoot because stem cells, then you think of like stem cells that we use in research. So shoot is better. Anyways, so if the solute potential decreases, then that means the water potential decreases. Yes, question.
Hi, I have a question about that we talking about the cell and the K moving is in the daylight or in the shadow.
Mm-hmm.
So we just in the notes I say we talking about is in the daylight, so it's will like close the summer and the cell and K will in, but why it's different? So I'm confused about this.
So you're saying in the daylight, what do you think should happen?
Oh, this is like we talking about is in the is this in the daylight or in the shadow
Shade.
shadow?
This is shade.
Oh, but we talking about in the notes we can see it's right down is in the daylight, so this will come in. So I'm confused about this. Why is different with that?
I think maybe what you're getting confused with is in the daylight, are you saying water comes into the guard cells
Uh,
or no?
uh, I'm not sure. Sorry.
So if you're talking about shade versus light, and you're talking about the shoot cells, then that's what we're talking about here. And then you would see water go in and elongation happen in the shade, but not in the daylight.
Oh, okay. Oh, I see. Thank you.
You're welcome. Was there another question?
The same. So talking all of a sudden about the manipulation of the water potential to bring solids in when we're talking about elongation during the shade.
Can you speak a little bit louder?
Yeah, I also had the same confusion about why we started talking about bringing solutes in and manipulating the water potential when we were talking about elongation during the shade?
Oh, so in order for cells to elongate, two things have to happen. There has to be water rushing into the cell, and there has to be the cell wall breaking up. When water rushes in and the cell wall isn't sturdy, that's going to cause the cell to elongate. So all of these steps to get solutes in are going to result in a lower water potential. And so water is going to want to rush into the cell in that direction. And that's one of the requirements of elongating a cell. You need water to rush in. And you can do that by lowering the solute potential. If that makes sense. Good? Maybe?
Yeah, that was great. Thanks so much.
Cool. All right, next question.
Hi, I had a question about the solute potential. So I know it is inverse, right? So it's like the solute potential is negative, right?
Yes.
Okay, so when we say like decrease the solute potential, that means it's getting more negative.
Yes. Yeah.
Okay, because I was like on the reviews and stuff, like once in a while, or even just me doing it, I would be like, oh, you're increasing that, like, I would do an increase in the solute potential. And then like in my mind, I'm like, oh, I'm increasing that negative number. But for the exams and stuff, would you guys prefer that we're saying decrease with that arrow? Like, or do we would just say like negative, more negative solute potential? Like as long as we just kind of show that it's getting lower, is that all you guys are kind of looking for?
Yes. You can do an arrow down or put more negative. Either one of those will work.
Okay, cool. Makes sense. Thank you. And I might be getting ahead here, but does the like the FOT, like the PHOT also have something to do with this? And would you possibly go over that as well?
Yes. Yeah.
Okay.
So PHOT is phototropin, which is another photoreceptor. And it gets confusing because phototropin in last unit did play a role in elongation when we are talking about how the shoot cells elongate to the opposite side of light. So last unit we said if there's light coming in from the side, it was actually phototropin that caused this exact same response in the cells opposite of light to make the plant bend towards the light.
Mhm.
Now we're saying when we're talking about shade versus light, not about moving towards the light, just shade versus daylight. Now it's phytochrome, and phytochrome deals with red and far red light, whereas phototropin deals with blue light. And this is the response to shade versus daylight.
Okay. So
Then
those two different things. Oh, sorry. No, keep going.
Last thing
You
I want to
answered
say,
it.
yeah, is phototropin then comes back this unit in the guard cells and is the reason why in daylight to blue light, the guard cells open.
Okay. Okay, there we go. There it was. Okay, cool. That makes a lot more sense. All right. Thank you.
Yeah, it's confusing. There's so many and they all kind of do such similar things, which makes it hard.
No, but that clarified a lot when it comes to the differences. So thank you. I appreciate
Yay. I'm glad. All right, next question.
So I have a really bad habit of writing phosphate when I'm trying to write phosphatase. And this is kind of just a semantic like thing, but like, is that fine or is she gonna be like picky about that?
So you put like sour inhibits phosphate.
Yeah.
I would say that's a case where I would be careful just because that would be doing the opposite effect, literally. If you stopped
Yeah.
phosphatase, that would be doing the opposite of what inhibiting phosphatase does. So I could see people maybe getting picky about that. So just, yeah, I would
Okay.
go with it.
Thank you.
You're welcome. Next question.
I had a question really quickly. Like for each of the flow diagrams, they usually let us only do it within like 5 steps. And so for something like this where there's multiple steps, how do we know which step to skip? Or like how do we know to understand like which ones to write when there's like 10 different parts to it and like what's important?
Great question. So my advice is to always include more. If you want to like combine some of the steps into to make it 5, you can, to where like you're saying lots of things in one step and then you move on, because you'll never get points off for putting too much, but you will get points off if you don't include the stuff we want you to include. So I would err on the side of putting more, and we would never take off and be like, oh, you did 6 steps instead of 5. So like that's supposed to give
Oh,
you an idea, but it seems like it messes y'all up and makes you say too little. So err
I
on the
think it's
side of
because
saying more.
on one of the tests she said that she's only going to check the first 5. Anything beyond 5 steps doesn't get counted. So you have to include everything in only 5 steps. I could be wrong, though. I'm pretty sure that's what she said. And so I think, yeah, that's like where I was having problems because I wasn't sure how much we're allowed to include in like one step.
Yeah. Anytime I've created a flow diagram, I haven't just read the first ones and then stopped reading. And I don't think the other TAs are doing that either. If you want to be careful, again, maybe do step 1 and then like write 3 different things in it.
Okay, perfect. Thank you.
You're welcome. Yeah, another question?
Hi, can you hear me?
Yeah.
Okay, I remember like the first day we covered this that she said that like shade avoidance is like a phenotypic expression. So does that mean only certain plants like demonstrate shade avoidance, and like should we just assume on the test that every plant is shade avoidant?
Yes. Yeah. I don't know of any that are not, but biology, of course, there are probably some that aren't. So, but on the test, yes. Assume that.
Okay, thank you.
Next question. Julia?
Yeah. I was just gonna ask, when you're done finishing that flow diagram, could you like zoom out so we could just take a picture of that?
Yeah.
Like the whole thing together? Okay, thank you.
Next question.
Yeah, so if the question says, explain this thing in 2 sentences, and I wrote like 5, will I get points taken off for that?
No. I think it's just like trying to make you not over explain. But if you're, I don't know, I would err on the side of saying more than saying less.
Okay, awesome. Thank you.
Yeah, no, we don't count sentences and stop reading after those 2 sentences. Maya.
So I got here kind of late, so sorry if you already answered like this type of question, but I just wanted to make sure if the length of the exam is like the same as the other exams, but we'll still have the full final time. Is that correct?
Yes.
Okay, thank you.
You're welcome.
So the exam will be only 5 questions, right?
I believe it's 6.
Okay.
Next question.
Hi.
Hi.
Can we go over any mass balance like diagrams that we need to know for the shade avoidance?
Yeah. No mass balance for this one.
Oh, okay, I see. Then if any mass balance needs to come up, can we go over that
Yes.
in like topic? Thank you.
Yeah. Okay, so those are the two flow diagrams. And then the last thing for shade avoidance to know is the trade-off that happens in the shade. So let's say we're in the shade, but also there is a caterpillar. Ooh. I wonder if like being on Zoom, it's just like too much for my Mac. Like can't take it. Okay, but anyways, shade plus a caterpillar is eating the plant. So the caterpillar is going to activate signaling cascades that result in an increase in the hormone JA. JA is going to go into the nucleus and bind its receptor in nucleus. It says nucleus, obviously. And then JA plus its receptor kicks off a repressor called JAZ. So kicks off. Ooh, JAZ. JAZ is a repressor of defense genes. So when JAZ gets kicked off, we're going to increase transcription of defense genes. Defense genes. If you need me to say out loud anything that I write, I will. It's a little bit laggy still. So that's what we learned last unit of how in response to herbivory, how the plant defends itself. It increases JA, that's the first signal, and downstream it makes defense genes that do various things to help the plant defend itself. But if it's in the shade, what we find is the PR form of phytochrome inhibits some part of this pathway downstream of JA. What is the result of that? That means that in the shade, we still see an increase in JA, but we do not see defense gene transcription increase. So it seems like there's a trade-off in the plant where it's like, do we want to put resources towards elongating, or do we want to put resources towards defending ourselves? And in the shade, it seems like they choose elongation and to not defend itself. So yeah, that's what you need to know about shade avoidance. And the last note I have here is it's going to be helpful that you can also read graphs. And specifically, what you need to know is if you have a graph that looks like this. Oh, I didn't do this very well, but oh well. I'm gonna do this. Okay, let's say you have a dot plot like this and you have some error bars. You're going to need to know that even though this dot is lower than this dot, because their error bars are overlapping, these are not different. This one, on the other hand, because the error bars are not overlapping with the other two, you can say with confidence that this is higher than both of these. But you could not say that these two are different. That's just a little gonna help you on the test. Okay, any questions about shade avoidance? I see one.
Where you say kicks off, you mean to say ubiquination degradation of that repressor? So is it okay to say kickoff instead of ubiquination degradation of sour or JAZ?
Yes, yeah. You could say removes jazz, kicks off jazz, causes degradation of jazz. Any of that would be good. Yes,
Okay,
question.
thank you.
You're welcome. Question?
How much overlapping would you consider to be like completely overlapping?
You could draw the lines out, like draw them. If they touch each other, that's overlapping. If they do not, it's not overlapping. Cool. So shade avoidance, you're thinking phytochrome, PR versus PFR stuff. And then also the fact that PR inhibits the defense pathway. Okay, now we can move on and talk about water movement through the plant. So basically transpiration stuff. So for that, the first thing I want to do, yeah, question. Someone raise their hand.
Yeah, I was wondering, so for when you say like understanding the graph, could you just quickly explain like what you would typically find on like the x-axis versus y and what the specific point means?
I feel like that's saying too much. I can't tell you literally like what the graph's going to be. I'm just saying it's going to be important to know what the error bars mean. You'll be able to figure out what the graph is showing you. That's not what you're being tested on, and it'll be clear. But I can't like say what's on the graph. Okay. Okay, so water movement through plants. I think what's really helpful when you're thinking about how water moves through a plant is not only knowing like at each level which part of the water potential equation is relevant, but also just kind of imagining the cohesion of the molecules of water. So like maybe there's a water molecule here at the very end. So it's like right on the edge of the leaf cell apoplast. And it's about to evaporate into water vapor. Well, that little water molecule is hydrogen bonding. It's showing cohesion with other water molecules. And those water molecules are bonded to other ones. And they're all attached from the leaf apoplast and that little water film on the leaf apoplast all the way through the xylem. So this is like the xylem tube going through the plant. They're all connected to each other via those cohesion hydrogen bonds. And they're connected all the way to the end, all the way into the root apoplast. And that's important because when this last water molecule here evaporates, that's going to cause a pull. And that pull is not just going to affect the next one, but every water molecule in that line. And what you should be imagining is that pull is an increase in negative pressure. And that causes a lot of problems for people because increase negative pressure is really confusing. So more pull because of more evaporation is more negative pressure potential. So the negative sign becoming more negative. That's how we measure that tension, which is that pull from the water at the top and all of the water molecules that are attached to it below via hydrogen bonds. Question?
Yeah, I think the reason why I get so confused with the whole negative pressure potential thing is because we never talked about like what the negative pressure potential means to begin with. So that's why I don't know why it would be negative.
Yeah. So what the negative pressure literally is it's a measure of the tension on water. So you can think of like positive pressure would be if I took a balloon and I blew air into that balloon, that would be positive pressure. If I took a full balloon and then I pressed on it, making the air go out, that's negative pressure. It's the same thing here. So when we have water rushing into a cell, we call that positive pressure, and that's turgor pressure of the plant. When we're actually kind of pulling water from the outside, from the top, that's negative pressure on the water. So kind of weird, but you have to somehow visualize it in your head. But the key is you should also think more tension. Oops, this doesn't look good. More tension equals more negative pressure. So becoming more negative, which would cause a more negative water potential. So the more and more tension there is, the more and more pull we're getting from up here, the more negative pressure in all of these cells, and that causes a lower water potential in all of those cells. So having that association in your head is going to be really important on the test. So you should just automatically think, if there's more transpiration happening, that's causing more negative pressure, that's lowering water potential. You should just like automatically go through that in your head. Less transpiration, you should think less negative pressure potential, less tension. So a higher water potential, a more positive water potential. Just like ingrain that relationship into your head. All right. Then we can talk about what are the parts of the water potential equation that influence water movement at each level. So we're going to start at the top and go down. Yeah, question.
Will this recording be on Zoom or on Canvas?
Yes, I will post it after.
Thank you.
You're welcome. So the water potential of the air or the atmosphere is just determined by the relative humidity potential. And basically you want to think that as temperatures go up, then the relative humidity and the relative humidity potential goes down. So if that goes down, that's going to cause the water potential to also go down. So what does that mean for water movement? Well, if the water potential of the air is lower, that's going to increase the gradient between the water potential of the atmosphere and the water potential of the leaf airspace. So that will increase flux of water. The other thing you should think of is what would happen if you have a high amount of rainfall on relative humidity. So that would cause an increase in the relative humidity of the atmosphere, which would increase the water potential of the atmosphere. So that would actually decrease the gradient between the two and cause less flux of water out into the atmosphere. Okay, and then next one is the water potential of the leaf airspace, which is also just relative humidity. And so the leaf airspace relative humidity is always pretty high compared to the atmosphere, just because if you were to like zoom in on the leaf airspace, you would see like a bunch of the crosslinks of the leaf cell apoplast, and along those crosslinks there is like a film of water. So if this is the leaf airspace, and this film of water is always causing evaporation of water molecules, it has a really high relative humidity, which is good because we want this one to be high compared to this one if we want transpiration and water movement to actually happen. The other thing that's going to be helpful for you to know is the relationship of the leaf airspace relative humidity and guard cell opening and closing. Guard cell open versus closed. So the main thing you need to know is when the guard cells are closed, that's going to impact the relative humidity of the leaf airspace by making it shoot up really high. So when guard cells are closed, the relative humidity of the leaf airspace is going to be near maximal levels. So because you're kind of preventing the water molecules from leaving the leaf airspace, when they evaporate, they're all staying in there, and it's gonna make the relative humidity in that area really high. So guard cells closed, really high relative humidity of the leaf airspace. Okay, next level is the water potential of the leaf apoplast. So that is right here in the cell wall of the leaf cells. And that is just going to be negative pressure. So that's easy. The only thing that influences that one is that pull, that tension. So let's kind of go into the details of what's happening there. So let's just say we're zooming in on the leaf apoplast cell wall, and they make a bunch of cross links. The cell wall parts look like this. And as you have low levels of transpiration or low amounts of evaporation, you're going to have more water kind of filling in these cross-linked areas. And if you have high levels of evaporation, then you're actually going to make that water level, that water film, much lower. Ah! That's not what I meant to do. Ah, that's not what I meant to do either. It's the delay. It's messing up my picture. Okay, this should be lower than, let's say, this one. All filled. So these two on the left, that would be like low transpiration, lots of water in the water film, the leaf apoplast. And this one over here would be like high transpiration. Yeah. Okay, so at each little cross-link, the water film's forming a meniscus or a menisci, right? And when there is low levels of transpiration, the radius of that menisci, like this one, is large. Right, if I were to continue the circle here, it'd be really big. Versus if you have high levels of transpiration, the radius of menisci is smaller. When the radius of menisci is smaller, that is more tension, which is more negative pressure. So over here, when I was drawing like a little water vapor molecule moving up and pulling and causing the whole chain of water to feel more pull. If we were to zoom in on that area, this is what's happening, and this is the physics explanation for why there's more pull. So if I were to ask you a question on the test, like, how is the negative pressure going to change in response to a high temperature environment? You wouldn't just say the negative pressure is going to increase. You would have to say, well, because the radius of menisci at the leaf apoplast has gotten smaller, there is therefore more tension and therefore more negative pressure, which means therefore a more negative water potential. So when you talk about an increased in tension, you want to bring it back to the radius of menisci in your explanation. Unfortunately, I feel like that's so much detail, but I'm just trying to tell you what you need to do. Okay, yes, Julian.
Yeah, what does LAF stand for?
What is what? LAF?
Yeah, it's the second line.
It's the leaf airspace. So if like this is the leaf apoplast,
Oh,
this is the leaf airspace.
so LAS, not LAF.
Yes, sorry. I don't know why I put F.
The white one. Tegan, Alec found this banger.
Someone needs to.
Oh my god.
Okay. Any more questions before I move on to the next level? All right. Then after the next level of leaf apoplast is the part that's kind of continuous with the leaf apoplast, which is the xylem. Water potential of the xylem. The xylem's dead. It's basically continuous with all of the apoplasts of this plant. So similarly, it's also basically determined by the negative pressure, which makes sense, right? How I said, if we have increased pull at the top, it doesn't just affect those water molecules there, but it's going to increase the pull, the tension on all of these here. So the water potential of the xylem here is largely determined by that pull, which we quantify as the negative pressure. The more pull, the more tension, the more negative pressure, which means the lower the water potential. Technically, also, gravity potential can influence the water potential of the xylem, but it's basically negligible in most cases, and it's going to be negligible on this test. Yeah, question.
Okay, so my question was, if you reduce the radius of the menisci, is that what's pulling and that's causing negative pressure? Is that what, oh my God, is that what is pulling the water from the xylem to the apoplast
Yes.
to like refill that film?
Yes. Yep.
Okay. Thank you.
You're welcome. Yes, another question.
I was wondering if you could really quickly just explain the difference between symplast and apoplast, because I always mix them up.
Yeah. So if this is a cell, the inside is the symplast, and the outside where the cell wall is the apoplast. So like this area, this apoplast area of the leaf cell is basically continuous with like the xylem. It's like a xylem tube, which is then continuous with the apoplast area of the root cells. So if there's water in all of these areas, then movement here is going to pull and cause more negative pressure for all of those areas. I don't know if that helps.
It does, yeah. Would the leaf airspace then be considered like apoplast or no?
No, because it's just airspace.
Okay, cool. Thank you.
You're welcome. Okay, so this one is definitely important for you to know. And what you really just need to know is that little relationship I've been trying to drill into your head. If there is high transpiration because there's hot temperatures or drought conditions or low rainfall, then that's going to cause, and I don't know how to write it in a way that's the least confusing, but I don't know, more negative pressure or decreasing pressure, more negative, smaller number. Smaller number here is going to cause a smaller, more negative water potential of the xylem. So this value should become smaller if there's more transpiration. Yes, question.
So on the lecture slides, I was talking about the different pressures involved on each of those levels. There was a distinction between positive pressure potential and negative pressure potential, and I was confused on what the difference is. I thought they were describing the same thing. You're just saying one is increasing, the other is decreasing, but you're talking about pressure potential. But on the slide, it was showing that there's a distinction between negative pressure and positive pressure potentials.
Yeah, so there's pressure potential, and there are different factors that influence it. More tension causes the pressure potential to become more negative, and that tension is a negative pressure. So more and more tension, more and more negative pressure the smaller and smaller and smaller the pressure potential is. Positive pressure is literally turgor pressure in a cell. The more water that rushes into a cell, the more positive that pressure potential becomes. Bigger and bigger and bigger number means a bigger and bigger and bigger water potential. So again, kind of imagine like the balloon example, if that maybe helps, right? Blowing air into a balloon versus squeezing it out. Squeezing it out is negative pressure. That's kind of what we do when we pull on water molecules. And when we rush water into a cell, that's more like blowing up the balloon. Question.
Yeah, could you explain how time of day affects water potential? Like in the xylem, for example?
Yes, that's a great question. So typically what we see is something like this. This is the water potential of the xylem, which for our purposes, we're just going to say is basically all about the negative. The negative pressure potential. And what we see is over the course of the day, something like this, typically. Meaning at the start of the day, we see the xylem water potential decrease. So what does that mean for what's happening with the negative pressure? If it's decreasing, that means we're getting more and more negative pressure early on in the day. Why? Because as we go through the morning, we open up stomates and we increase our transpiration. So if we increase our transpiration, we're increasing that pull from the top, and that pull is more and more negative pressure. More and more negative pressure means the water potential goes down. Then it hits some kind of maximum low rate of water potential because we have all of the stomates open. We're making water go through as much as possible. We have kind of maximum tension, maximum pull, maximum negative pressure. And when I say maximum negative pressure, I mean it's really low, right? It's going from like maybe negative 0.8 to negative 0.10. It starts off at negative 0.8 and then becomes more negative. Sorry, I mean negative 0.08, right? That would be more negative. Yeah, sorry. 80 to 10 would be the opposite direction. And then as you get to the evening, then the stomates are going to close back up again. So as the stomates close, there's less transpiration. So there's less pull, less tension. So there's less negative pressure. If there's less negative pressure, then the water potential will rise. It will become less negative because there's less negative pressure. So I'd say maybe that's like dawn. This would be like dusk. Oops. Maybe 12 p.m.
Okay, yeah, that helps a lot. Thank you.
Good, I'm glad. All right. So that's the xylem. And then the xylem, the next level would be the roots. So you could think about the water potential of the root. Oops. Apoplast. And then also just the root cells, the live cells.
And then that's the next question.
Okay, so the water potential of the root apoplast is again just the negative pressure. That's why it's like a whole chain of water molecules. And again, this is influenced by pull from the top. As more transpiration happens, this becomes more negative. The root cells are also going to be influenced by pressure potential, but now it's positive because it's a live cell, not the apoplast. So inside a live cell, you don't get pulling on water. If you did, the cell would collapse. So if the cell is at least alive and has some amount of turgor pressure, then it's going to have a positive pressure, and that's the pressure of the water pushing out against the cell wall. I mean, you could manipulate the cell in an experiment to have it have negative pressure, but you would kill it. Okay, so it's positive pressure and then also solute potential, which is what y'all learned last unit. Yeah, question.
Does that mean that the leaf rod cell have the pole of itself? It's not on the top, just the itself have the pole, so it's a positive. Does that mean or not?
The positive pressure just refers to the turgor pressure of the cell. And turgor pressure is the water inside the cell is pushing up against the cell wall from the inside. Like,
oh, okay, yeah.
Pressure inside the cell against the cell wall. That's positive pressure. And you can imagine if you had like a pull on the water molecules from the inside, you would like make the cell kind of shrivel. That would be negative pressure. Blowing up your balloon like here, that's positive pressure. So we don't have negative pressure inside cells if they're living. We only get negative pressure in the xylem and in the apoplast. And it's specifically due to that tension, that pull. Okay, and then the last level that you can think about is the water potential of the soil. And the water potential of the soil is also influenced by the solute potential and negative pressure again. But this time, this is a very different negative pressure than the ones I've been talking about before. So the negative pressure in the soil doesn't primarily come from a pull from the top. It's not really directly connected to that full chain. It actually has to do with the water levels of the soil. And it's kind of synonymous to what we saw in the leaf apoplast. So in the leaf apoplast, we talked about how depending on how much water was there, it changes the radius of menisci. A similar thing happens in the soil. So if I had like some soil particles, oops. And there was like a ton of water in the soil, then it would look something like this. And if there was a drought, very low rainfall, not much, then it would, the menisci of the water in the soil would look something like that. So you can see in high water, lots of water in the soil, you have a large radius of menisci, and in low water in the soil, you have a smaller radius. What does that mean? It means small radius equals more tension. And again, it's that same relationship you should have drilled into your head by now. If there's more tension, then there's more negative pressure. That's very negative value for this, which means very negative value for water potential. So what does that mean for water movement from the soil? Well, if you make the soil water potential super low, and water likes to move from high to low, it's not going to want to leave the soil as much. If this gets lower and lower and lower, we've decreased the gradient from the soil to the root cells. So we get less movement of water into plants in low water soil, which is intuitive, but again, on the test, you can't just say like, obviously it's dry soil, less water movement. You're gonna have to say this. This is what you have to say in 220 for why that's true. You say smaller radius of menisci between soil molecules, or sorry, soil particles, not molecules. They're particles, multiple molecules put together. Smaller radius of menisci, more tension, more negative pressure, lower water potential, smaller gradient of water potential in the soil versus the root cell, so less flux of water out of the soil. All right, questions on that? The different water potential values at each level. All right, I'm going to go to the bathroom really quick because I have to pee really bad, and then I will be back. Okay, sorry, I'm back. Let me share my screen. Okay, question.
Yeah, hi, can you hear me?
I can.
Okay, so just for clarification, you would say more tension between the soil particles when there's lower water in the soil?
Yes.
Okay, cool, thank you.
You're welcome. Okay, next thing I want to do is just write specifically what relationships you should know about this stuff. So the first one, I want to go back to black. Okay, temperature. Ooh. Temperature. No, my flips is back on. I wonder if y'all can see that. Okay. Temperature. So you should be able to talk about how water movement's going to change to increasing or decreasing temperature. So let's just say it was increasing temperature. The effects of that would be, on one hand, to lower the relative humidity of the atmosphere. So that would lower the relative humidity potential and therefore lower the water potential of the atmosphere. So then it would be higher gradient. Oops. Higher gradient between the water potential of the atmosphere and the water potential of the leaf air space, LAS. So higher transpiration. And then more negative pressure. So if there's more negative pressure, you would expect a decrease in the water potential of the leaf apoplast and the xylem and the root apoplast. That's going to cause a pull all the way through the plant. All right, the other effect of increasing temperature, you could say, would be decreased water in soil. If there is decreased water in soil, you should imagine something like this: very low levels of water, smaller radius menisci. Yes. Radius of menisci. Less tension. Less negative pressure. Ugh. Which means a higher water potential of the soil, which means a smaller gradient. Between the soil and the root cells. Oh, this is rough. Between soil, water potential, and root water potential. All right. Yes, question.
So I thought that increasing or decreasing the radius, making the radius smaller would actually increase tension, not decrease it.
Whoops.
It says it'd be less tension.
Yes, so sorry. Yeah, sorry. I just got messed up. Okay. So decreased radius of menisci would be, that would actually cause more pull between the water molecules. Sorry, y'all. My brain is like going on autopilot. So that would be more tension, which would be more negative pressure. So if there's more negative pressure, then the water potential will decrease. And if the water potential decreases, then there's a smaller gradient between the soil water potential and the root water potential. So if this water potential value of the soil was really high, then that would cause a larger gradient, a steep difference between the water potential of the soil and the root, which would cause more flux in. But in the case of low water in the soil, by decreasing the water potential of the soil, then we decrease the gradient and make it harder for water to come in. Sorry. So in the case of high temperatures, we get increased transpiration and more pull from the top. And we also get the case of less water coming in from the bottom. If you did like a mass balance of that, the rate in from the roots would be going down, whereas the rate out from transpiration would be going up. And you get a decrease of water in the plant. And if this becomes too big relative to the rate in, then that pull from the top can actually break that chain of water molecules. And when it breaks the chain, it creates a little air bubble in the xylem known as cavitation. Yes, question.
Yes, so wouldn't the gradient be bigger? Because doesn't it go from high to lows? Or am I
Yeah.
for like the last part of this flow diagram?
Yeah, so if we had like soil and root, in order to get movement of water this way, we would want this to be really high water potential. Ugh, this to be really low. So if we decrease this, we're decreasing the gradient. If we make this really high, 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 we increase the gradient.
Oh, okay, I understand now. Thank you.
You're welcome. But this is a good time to think, you know, I just messed this up, which shows you, like, if you have not had enough sleep tomorrow, even if you know this stuff, you might put the exact wrong thing. So make sure you get enough sleep. All right. Questions about this? Okay, so the other one that you should know for sure is, and I'm just gonna kind of do it with this one, is rain / wet weather. Yeah, question?
I had a question about ABBA. Does that come into this kind of
Yes.
genre?
Yeah.
Okay.
So I'll talk about that right after this one. I just want to say these two
cool.
things. Okay, so rain, you're welcome. Rain, wet weather. If there's rain, it's going to do the exact opposite of increased temperature. Rain is going to increase the relative humidity. And if you increase the relative humidity of the atmosphere, that will increase the water potential of the atmosphere. And then you create a lower gradient. Right? Again, with that same explanation, where if you want water to move high to low, if you make the low part too high, then you've decreased the gradient. You've made the difference a lot less different. So that's going to decrease transpiration when there's wet weather. If there's decreased transpiration, you should think less pull. Less pull means less negative pressure. So if there's less negative pressure, then you should expect the water potential of all of these things to go up. So not only should you know, well, I'll say that in a second. I'll just go over to this, the other side, and then I'll say what I was going to say. So again, if we think lots of rain, then it's the opposite effect. The water in the soil should increase. If there's more water in the soil, you should imagine this kind of all filled up, and that radius of menisci is a lot bigger. If it's bigger, that's actually less tension on these water molecules. There's less pull down. So that would be less tension. Less negative pressure. So a higher water potential of the soil. And if you make the water potential of the soil really high, now we've increased the gradient from the soil to the root. We've made it steeper. So that should cause more flux into the root. So yeah, when you think about water movement, it'll just be helpful on the test tomorrow to remember the temperature effect on relative humidity, water and soil, and then rain / wet weather effect on relative humidity and water in the soil. And what's fair game is I could give you a graph of these things, and you should be able to tell me which graph would be a higher versus low temperature, or which graph shows more or less rain. What would I be showing you on the graph? Maybe a change in the amount of water in the soil, or a change in the size of the radius of menisci, or I could show you on the graph a change in the water potential of the leaf apoplast or the xylem or the root. And given that you know these flow diagrams, if you see like the root apoplast water potential, one line up here and one down here, you should be able to tell me, okay, the lower one, the lower water potential, that must be the case of high temperature or not much rain, because I know that this water potential drops in the case of high temperature or low rain. So if you get confused on the test, maybe kind of write out this flow diagram of the two things you expect, and that will help you if you're given a graph of this stuff. Cool. Let me see if there's anything else I wanted to say. No, not on that. Okay, so then we can think about what happens in the specific case of like a prolonged drought. Drought effects on water flow. So the first thing is going to be high transpiration. Because of, what we just said here, right? In a drought, there's both high temperatures and low rainfall. The other thing that's going to happen is low root water uptake. Because of those two things, we're going to see cavitation. So if we have our little plant, our little chain of water molecules, if the rate out gets really high, but the rate in is pretty low, then we can actually end up breaking the hydrogen bonds of that chain of water. And that causes an air bubble, and it's a big problem because now if we increase transpiration, we're not going to pull up an entire water column because now it's separated. So we're not going to get enough water moving through the plant, and that can cause plant death. Then another thing that's going to happen is plant stress response. So it will detect in the roots that there's less water coming in from the soil. And in the roots, the response is going to be ABA synthesis. And ABA is going to travel up to the guard cells and cause the guard cells to close stomates. So this is a drought response because we can see that this high transpiration is causing problems for the plant, like cavitation. And this ABA response tries to reduce transpiration and prevent some of those things like cavitation from making the plant die. Then what we might see is reduced defense response. Of the plants to herbivory. Why would that be? So it kind of has to do with what ABA does. Oops. Ah. No, I don't want that. I don't want that either. Go away. Stop. So, oh my gosh, what happened? Why am I drawing squares? I just want my pen back. Okay. ABA is going to close stomates. Which is going to decrease CO2 intake. If CO2 can't come in, there cannot be photosynthesis. There can't be carbon fixation. And if there's no carbon fixation, there is no G3P. And if there's no G3P, then there is no sugars for doing cellular respiration. And if there's no sugars for doing cellular respiration, there is no ATP. And that's a problem because you need ATP to synthesize defense molecules. So if you have less energy, you're going to defend yourself less. Yeah, so that kind of gets at the three hypotheses of the responses to drought. And you're not going to have to list those hypotheses, but you're going to have to understand what you would expect to happen under the case of drought, which would be these things. High transpiration, low root water uptake, which could cause cavitation. Then you would expect an ABA response to close stomates, and that would reduce CO2 and also reduce energy for making defense molecules. All right, questions about that? Yes.
Is G3P made in cellular respiration or photosynthesis? Because I thought it was photosynthesis.
Yeah, it's made in photosynthesis, and then cellular respiration takes 2 molecules of G3P, glucose, and breaks it down for energy. So G3P gets used as a reactant kind of in cellular respiration. All right. Let's do the next topic, which is how and why do plants open and close their stomates? And there's 2 relationships you need to know about stomate opening and closing. So one is to day versus night. So they open their stomates in the day, oops, and close them at night. Go away. Ugh. Ugh. And the other one is ABA. So basically, you can't just say, oh, daytime's going to open them and ABA is going to close them. You should know the full flow diagram of how those 2 things affect or cause the stomates to open and close. And they do so by changing the amount of water in the guard cells. So when the guard cells have a ton of water in them, meaning they have high turgor pressure, high positive pressure, then they're open. And when they have low water in them, then they're closed. So ABA is going to cause water to move out to close the stomates, and daylight is going to cause water to move in to open the stomates. So this signaling has to do with blue light, which means phototropin is involved because phototropin detects blue light whereas fight detects red and far red light. Okay, so in response to blue light, it's going to cause opening of stomates. And how it's going to accomplish getting water to want to move in there is by manipulating the solute concentration. So what happens? So blue light is going to be absorbed. Absorbed by phote. I want that line to go away. Okay, and then signal cascade. And then we get increased activation of a kinase protein. And kinase is the one that adds phosphate groups. So the hydrogen pump needs a phosphate to activate. So more kinase means more phosphate added to the hydrogen pump. So that means increased hydrogen pump activity. Yes, question.
Yeah, is this kinase like directly like next to the hydrogen pump where ATP is, or is it like somewhere separate?
No, yeah, I mean, this is all happening in the cytoplasm, but then the kinase acts on the pump in the membrane.
Okay, thank you.
You're welcome. Okay, so kind of the big difference between like shade elongation and what's happening here. Notice here we have phote. This is all happening in the guard cells, not the shoot cells. So it's phote instead of fight. We're not doing red light, so we're not doing PR or PFR. We're affecting a kinase, not a phosphatase. And there's nothing involved in the nucleus. We just kind of directly act on the kinase and then hydrogen pump activity. But everything kind of downstream of that is the same. So that's going to increase the hydrogen electrochemical gradient across the membrane, right? We have a plant cell. We have a low concentration now inside and a high concentration out. We can use that gradient to pump in or transport in chloride ions. So the hydrogen chloride symporter is going to bring chloride in, just like before. What else is it going to do? It hyperpolarizes the cell. Because positive ions leaving means the membrane potential becomes more negative, or it hyperpolarizes. That will activate hyperpolarization gated potassium channels. And it just so happens that at that membrane potential, the electrochemical gradient for potassium is inward too. So potassium will move in. Then we have an increase in solute concentration, which is a decrease in solute potential, which is a decrease in water potential inside the guard cell. So that's going to cause water to move into the guard cell. You put increased gradient. Flux. into cell. Ugh. Into cell. So that is how daylight opens the stomates. Okay, and then how does ABBA work? So ABBA gets synthesized in roots. Travels through the xylem. Through. To guard cells in the leaf. This is also going to be ABBA in the cytoplasm. So ABBA binds receptor in the cytoplasm. Binds receptor in cytoplasm. And ABBA, ABBA plus its receptor is going to directly activate a voltage gated chloride channel. Oh, why is this not erasing? Okay, we're just gonna ignore that. So ABBA is going to activate a voltage gated chloride channel. And it just so happens that at this membrane potential, when those channels open, chloride moves out. So note, we don't have that increased hydrogen pump activity. So we haven't made the membrane potential super low. So the membrane potential right now, normal conditions when ABBA's around, it's going to be more depolarized than it is over here. So the chloride ECG is actually out. The other thing it's going to do is it's going to actively inhibit the hydrogen pump. So not only do we not activate the pumps and hyperpolarize the cell, but we actively turn them off. So that's going to depolarize the cell. And if we depolarize the cell, we're going to activate voltage gated potassium channels. And it just so happens that at that membrane potential, the electrochemical gradient is for potassium to move out. So you can see the difference in the two flow diagrams. When you have hydrogen pump activity, that's going to hyperpolarize the cell, and the electrochemical gradient for these ions is in. Over here, when you inhibit the hydrogen pump, you depolarize the cell, and now those ions actually want to both move out. So if you remind yourself, how do you know if an ion wants to move in or out? That's all about the electrochemical gradient that you calculate by using the Nernst equation and the electrical gradient to get the overall electrochemical gradient. So that is going to be a thing you will do on this test for sure. Okay, but if we have ions moving out, then the solute concentration decreases. So the solute potential increases. So the water potential increases, which means you have a decreased gradient and a decreased flux of water into the cell. So you get the stomates closed. Question?
Yeah, so I thought it was a given that ABBA makes it so that ions move out instead of them coming in without ABBA. So why would we have to calculate like the membrane potential since we already know like the end result?
So you just might be given a case where it's like, under normal conditions, ABBA does this. But in this cell, there's a weird mutation, and for some reason, the concentration of potassium is now this in and out. How would that change the response to ABBA? So you would have to calculate what is the actual potassium ECG. Is it in or out? Because even if you activate ABBA and you open these channels, the only thing determining which way potassium moves is that ECG. So you'll be
Thank you,
yeah, in some kind of weird case. Okay. Oh, last thing I wanted to say is you should know the relationship between open and closed stomates and these 4 things. Number 1. Actually, 5 things. I forgot. I just added it on, but I already mentioned it. So number 1, carbon fixation. If you close stomates, less carbon fixation. Plant temperature. If you close stomates, the plant heats up. Why? Because there is less evaporative cooling. Guard cell turgor pressure. If you close stomates, that's less positive pressure, less turgor pressure. 4, xylem water potential. You close the stomates, that's less transpiration. So the water potential of xylem goes up because there's less negative pressure. And then the 5th one is leaf airspace. If you close the stomates, leaf airspace, relative humidity shoots up to almost max. So its water potential also goes up. So know those 5 relationships to open and closed stomates. Cool. Yeah, question.
Do we need to know the flow diagram for how stomates close at nighttime? Like, is that influenced by FOT just like not detecting blue light and then it just slowly becomes more flaccid, or is it
Yeah, it would
having
just
to
be
do with
the exact same flow diagram in the opposite direction. No blue light absorbed by FOT, no signal cascade, less kinase, less hydrogen pump activity, less solutes going in. That would be that night.
Oh, okay, thank you.
You're welcome. Okay, the last thing I wanted to do, and we kind of already talked about it, but might as well just be more specific about it. You should know the xylem water potential over time graph. Over time. So let's say this is graphing water potential of the xylem over the course of 1 day. So maybe this is like 5 a.m. This is like 12 p.m. This is like 6 p.m. I'd see something like this. And you should be able to interpret how the fluxes change as the water potential graph changes over time. So what are the fluxes? One of them is transpiration rate. So I'll do that in red or orange-ish red, I don't know. So how's transpiration rate going to change over the course of a day? And how can you tell from what's going on with the xylem water potential? Well, remember, as the xylem water potential goes down, that means there's more tension, more negative pressure. What's the source of that? More transpiration. So I know that the transpiration rate must be going up all throughout the region that the xylem water potential is going down. So it should look exactly opposite. And then when the xylem water potential is not going up and not going down, the transpiration rate is at its maximum. So what's happening here as we're increasing transpiration rate is we are opening stomates. And then at this point, all of our stomates have opened. And then when we see the water potential go back up, meaning there's less negative pressure, less tension, I know the transpiration rate has gone back down. And the other flux is the root water uptake. And for this one, I think it's helpful to think of the root uptake into the apoplast, because the root apoplast is continuous with the xylem, right? So as the negative pressure becomes more negative, as this becomes more negative, I know there's also more pull in the water of the root apoplast. And if there's more pull there, what does that mean? That means that a root apoplast water potential is decreasing. So as this decreases, this also decreases. How can that help me determine the rate? Well, if we're comparing the soil water potential to the root apoplast water potential, and we want water to move in this direction, if you make this lower, you're going to increase the rate, right? Because you're increasing the gradient. So as the xylem water potential is lowering, I know this is also lowering, and the rate of root uptake should also increase. Now I want to think, is it going to increase like this? Or is it going to increase like this? Or is it going to increase like exactly like the transpiration rate? Well, the xylem curve can help you with that too. If the root uptake was larger, if it was higher than the transpiration rate, then the water potential of the xylem would not go down. right? You need a higher pull from the top in order to get more negative pressure to get that xylem water potential to go down. So I know it can't be larger. If it was exactly the same rate in and out, then you actually wouldn't get a change in the xylem water potential, right? The only way it can become more negative is if there's more tension, there's more pull from the top. So then I can conclude that it has to be also increasing, but it has to be below the transpiration rate. And then once the xylem water potential is flat, so should draw it, I'll just be more precise just because. Once it becomes flat, the rate in and out should be the same. And then when the xylem water potential is increasing, that's less and less tension happening. And part of the reason for that is because there's less and less pull from the top, and also because the rate in is exceeding the rate out. So big picture to kind of remember what that looks like is the rate of transpiration and the root uptake. They almost look like mirror images of the xylem water potential. And then the root uptake is just a slightly delayed version of the transpiration rate. That can kind of help you remember it. Yeah, question.
Is low the transpiration. If it's low, doesn't water want to go in then? So then why would transpiration be there?
Oh, I didn't really hear you. Could you try talking louder?
Oh, yeah. I had a question about the, wait, can you hear me now?
Yes.
Okay, yeah, I had a question. If the water potential is low, then why would the transpiration rate be high? Because if water potential is low, then wouldn't we want more water to be entering?
Yeah, so why is water potential low when transpiration rate is high?
Yeah, like why is transpiration if water potential is low?
Yeah, so remember we said increased transpiration equals increased tension, right? If you imagine like the little chain of waters, as you pull more from the top, you're increasing the pull, which is an increase in tension, which is an increase in negative pressure. So an increase in negative
So adding more negative.
Yes. More negative pressure. Yeah. So that equals a lower water potential of the xylem, which is mainly determined by negative pressure. So this being high makes sense for this to be low.
What does it mean by negative, like more negative pressure? Like if you, so I get like how transpiration is increasing the tension, but then how does that add more negative?
That is just how we quantify more tension. We quantify it as a more negative number. More tension is a change in negative.
Okay, so then what's receiving, or like the negative pressure is occurring like in the xylem? We just should know that more negative pressure is occurring in the xylem, which means more tension.
Yes. Like the most helpful way in my head that I use it is imagining that pull from the top. And the more pull, I just in my head think that's a more negative pressure. More and more negative, less and less and less, lower, lower, lower. water potential. Like you just have to associate those two things. Hopefully that helps. Okay. Okay, that's it for water movement. So then we can move on to nutrient uptake, which I'm super excited about. When I saw the exam for the first time, I thought the nutrient stuff was extremely difficult. And I told her so, and it's been changed to something that I think is very fair. So I'm excited. I'm back to being excited about nutrient uptake. So the next unit was nutrient uptake. And the nutrients that we focused on was carbon from the air, and also anions and cations from the roots. Okay, this is the one place I would say you need to be prepared for a mass balance question. And it has to do with the carbon nutrient uptake. So first of all, I want you to star that and remind yourself that most of the plant biomass is from carbon in the air. And the mass balance you're going to need to know is that if we think about carbon in the plant or biomass of the plant, you should know the rate in and the rate out. And if I change the rate in or out, you should be able to draw your arrow of increased or decreased biomass in the plant. So what are the rate in and out? The rate in is carbon fixation. In photosynthesis. And the rate out is cellular respiration. So when you're doing a mass balance, here's what is literally graded. We grade that you label the container correctly. So make sure whatever the container that you're increasing or decreasing in, you label that. Then you want to put what is the substance in the container. That's another point. Then you get a point for correctly identifying the rate in and out. Then there's another point for have you changed the arrows the way you would expect in the situation given, right? Is this one bigger or is this one bigger? And then as a result of those changes, have you changed the arrow inside the container? So if you have an increased rate in, you should also draw an arrow going up inside the container. So if I said something that increased photosynthesis, this would be a full points answer. And I think a lot of times you might just forget, or you think it's obvious that this should go up if you drew a bigger arrow here, but You have to draw all the arrows to get all the credit. Okay. And then what do you need to know about nutrient uptake of anions and cations from the roots? So you should know kind of how is this done normally? Oh my goodness. Normally versus in infertile soil. Cool. Okay, so let's go over how it's normally done with the example of potassium. So if there are just normal amounts of potassium in the soil versus the root cell, I'm just going to put some numbers on this because I think it would be good practice for everyone. So let me go to the actual numbers. Okay. Let's say the concentration is 5 millimolar out and 150 millimolar in. So first, what you should feel comfortable doing. Oops, actually that's wrong. Sorry, that was an animal cell. 101 is more physiological for plants. 101. So first thing you should feel comfortable doing is calculating the ECG. So let's say, actually no, I'm just going to say, yeah, calculate potassium ECG. And you will be given the Nernst equation being equilibrium potential equals 59 over Z log out over in. So how would you calculate the ECG? So the Nernst equation gets you the equilibrium potential for the ion, which is also the concentration gradient. So it would be 59 over 1, because that's the charge of the ion, log out over in. All right, you guys do the calculation with me in case I do it wrong, because I've been known to do math wrong. Okay, so Log on here. Yes. No. Oh. Okay, did you guys get negative 118? Please say yes. Math is hard.
Yes.
Yay, I did math right. It's exciting. The amount of times I've done math wrong in front of large amounts of people is not good. Okay, so that gets you the E equilibrium potential or the concentration gradient. So what is the ECG? So what I kind of like to do whenever I'm doing this is imagine that I'm a little potassium ion in the cell, or maybe out of the cell. But either way, as a potassium ion, I want the membrane potential to equal to 118. So if the actual membrane potential is more positive, so let's say it's negative 110, then as a potassium ion, I want to move myself with my positive charge in order to get the membrane potential to this value. So I want it to become more negative if this is where we're at. So if I want the cell to be more negative and I'm positive, then I want to leave because that's going to make the membrane potential more negative. So the ECG would be when the membrane potential is larger than negative 118, then the ECG is inward. I mean, sorry, outward. And then let's say if the membrane potential was like negative 200. Now it's way more negative than I want it to be. I want it to be here. So as a potassium ion, how can I move to make the membrane potential go from here to that value? Well, I want it to be more positive. So I would now want to move in. So now I know the ECG is whenever the membrane potential is less than or more negative than negative 118, then as potassium, I want to move in. And this makes sense if you think about what I told you the hydrogen pump does. So let's say the hydrogen pump makes the membrane potential negative 135 millivolts. Then at this value, we have potassium moving in. Whereas if we inhibit the hydrogen pump activity, and let's say the membrane potential isn't so negative anymore, maybe it's like negative 110, well, now potassium wants to actually move out. So this is what I'm getting at when I say there's nothing special about the hyperpolarization gated channel or the voltage gated channels causing potassium to move one way or the other. It all has to do with whatever the ECG is. And you know the ECG by calculating this value. And then knowing the direction, either based on doing the EG minus CG or CG minus EG, depending on what makes more sense for your head, or doing this kind of thought experiment of like, well, here's where I want to be. how can I move as the ion to get it there? So just make sure you can do those calculations. But I diverged when I did all this calculation. We were talking about how does potassium get into the root cells in normal conditions. So normal conditions, this is what the ECG is. And we actually have hydrogen pump activity. Yeah, Prisha.
For the -110 and -200, why do both the arrows go in? Or were you just like trying to indicate?
Yeah, I was just.
Wouldn't that? Oh, okay. I just wanted to make sure.
Yeah.
And then the -110 and 200, are you just basically saying that we can come up with any number that's greater than -118 or less than -118? Or like, how did you come up with the numbers -110 and -200?
Yeah, I just used these as a thought experiment for getting these
Okay.
rates.
Okay, and so then on the test, it would be like what would we be given in order to figure out whether it needs to be looked in?
That's a great question. And I will come back to it really quickly. I just want to say a few things and then I'll come back to that.
Okay, yeah, go, yeah.
Okay, so under normal conditions, how do we get potassium into the roots? The hydrogen pump is working. And because of that, we have this very negative membrane potential, right? It hyperpolarizes the cell. And then that activates the hyperpolarization gated potassium channels. And there's nothing special about those channels, but we know at this very negative membrane potential, potassium under normal conditions, if there's normal concentrations in and out, potassium will move in. Okay, so now let's think about what would happen under non-normal conditions. So let's say that potassium concentration in the soil is super low. So before it was 1 out and 100 in. Let's say it becomes like 0.001 out and 100 in. What is the difference? Again, I'm going to have you do the math now. That way you're not doing it for the first time tomorrow. So now you would get a question like, okay, here are the normal concentrations. Here are the concentrations under a nutrient depleted soil. What is going to change? And use math to support your answer. So what would you do? So first you would find what is the new equilibrium potential for potassium. So it would be same thing, but now we've changed the out concentration. All right, everyone should do the math with me. That way tomorrow you're like, I feel comfortable. I've done this already. This is so easy.
Are these the real numbers?
I mean, these aren't the numbers that are going to be on the test. That would be bad. That would be no challenge at all. But you're going to get normal soil concentrations, and then they're going to be changed in some way. I'm not going to tell you the actual numbers or in what direction they're going to change, but they're going to change. Okay. Okay, now it's 295. Anyone else get that?
Yep.
Yay! Cool. All right, what does this number mean? We've changed the ECG, right? So now the ECG is whenever the membrane potential is greater than negative 295 millivolts, potassium is going to want to move out. And whenever the membrane potential is less than 295 or more negative than, it's going to want to move in. Okay, so that's the math you would have to do to support your answer, but what is the effect of this? Well, think about what we just said normally happens. Hydrogen pump is active, gets it to like negative 135. Okay, now when we're at negative 135, which way does potassium want to move? It no longer wants to move in at that value, right? Because negative 135 is a more positive number. It's a greater number than negative 295. Potassium actually wants to move out. So this is the big problem. When you get low nutrient soil, if you kind of activate this normal signaling pathway, you're going to open up these hyperpolarization gated channels, but they're going to actually let potassium out. So in the low nutrient soil, not only are you taking in less, you're actively pushing potassium out. So that's a big problem. So yeah, that is the math you should be prepared to do. You should be prepared to calculate electrochemical gradients, and then tell me how is that going to change ion movement from root to soil, given different changes you might see. Now you should also know what is the plant's response to this? Do they just die, or do they have some way to try to compensate for low nutrient soils? And they do. They have some tricks up their sleeve for dealing with this. So in low nutrient soil, plants will increase transcription of transporters. Why is my flux going crazy? Disable. Great, this is awesome. This solves all the problems. Why? So transporters could mean, for example, a hydrogen potassium symporter. So remember, that's something that uses the concentration gradient of hydrogens to actively pump in this other ion. So this is great because if we go back here, hydrogen pump is on. These channels are now useless. But if we start to make these symporters, we can still get potassium in, even though potassium's electrochemical gradient is crazy. So this is how plants compensate for low nutrient soil. And you should be prepared for, I've given the example of potassium, but this could be any cation or anion. So it could be chloride, could be nitrate, could be magnesium. And the other thing you should know is this little regulatory mechanism that plants use in response to low nutrient soil isn't one directional. So if there's low nutrients, plants will increase transcription. If there's high nutrients, they can also decrease transcription. So they will respond with the opposite thing of whatever's going on, if that makes sense. Yeah, question.
Yeah, I had a question about making those symporters. So even if you do have a lot of like co-transporters available, wouldn't it not matter because the electrochemical gradient of potassium is still like against whatever the symporter is trying to do? So it wouldn't matter if they're open for potassium to come in because the electrochemical gradient isn't for it.
No, actually, because this is active transport. So it's not just a channel. We're using hydrogens to actively pump potassium against its concentration gradient.
Oh, that makes the whole difference. Okay, thank you.
You're welcome. Yeah, other question?
So when we're trying to find the electrochemical gradient, don't we need to know the membrane potential? Because the Nernst equation only gives us concentration gradient, right?
Yes, but you can determine the electrochemical gradient as ranges if you're not given the membrane potential. And sometimes that can be hard to do, and that's why I, for example, gave myself some numbers. So I could think, okay, if it's above or below this number, what is the ion going to do?
Oh, okay.
And the other kind of relationship that you should notice here is like, if you're in the situation of a low nutrient soil, you can see what's happening in the math is that this number becomes more negative. If you were dealing with a high nutrient soil, this number going up would make this more positive. That could be helpful too. Yeah, another question?
You found those like values, those are on our reference sheet, right? Like what it would happen in normal conditions.
Yes, yeah. So yeah,
Okay.
the first concentrations I used was from the equation sheet. And then the
Okay,
change
so
one.
if we were given, okay, like any kind of question that we kind of, they don't give us a resting like membrane potential, we can just assume it's under normal conditions, which are on the reference sheet.
Yes, yeah.
Okay, sounds good. All right, thank you.
You're welcome. Cool. Yeah, I think that is it for nutrient transport. Oh, okay. Last thing I just want to mention that could be helpful is notice that this nice little response they come up with, which is regulating up and down the number of transporters. This only works if you have working hydrogen pumps. Meaning, if you block the hydrogen pumps, or you see something about messing with the hydrogen pumps, the plant's ability to deal with low nutrient or high nutrient or messing with the nutrient soil is going to be messed up. Because even if they make these new transporters or they downregulate the transporters, if you don't have the hydrogen pump activity, then this isn't going to work. So that's just a little note. Okay. Last topic is photoperiodism in flowering. And then we're finished. Okay, so for this, you want to be able to say, given an experiment, can you tell me if flowering will occur or not? So when does flowering occur in plants? So it has to do with the relationship between a CO protein, and honestly, this is so oversimplified. So if you're like really into this stuff, know that this is very much not exactly how it works. But for this test, this is how we're saying it works. There is a protein called a Constance protein, and the Constance protein can do one of two things. It can act as a repressor of the Florigen gene, or it can act as an activator of the Florigen gene. And the Florigen gene codes for genes that cause flowering. So what do I mean? This CO protein, this Constance protein, does exactly opposite things. When does it do which of those things? It acts as an activator in long day plants. And it acts as a repressor in short day plants. So the key idea is that if you have this Constance protein around, which is not all the time, it has to be stabilized, you'll get these two downstream effects in the plants, depending on if they're long day plants or short day plants. Now, the other key thing to know is this FT gene that codes for things that make the plant flower is going to code for a hormone that can move throughout plants. And that's just important because if you have like a long day plant, say this is long day, and you graft onto it a short day plant, and let's say you create conditions where there is CO protein around, then the CO protein is going to inhibit the FT, the Florigen, in the short day plant, but it's going to activate the FT in the long day plant. Because FT is a hormone that can travel, if it's made down here, it can travel up to the grafted on short day plant. Meaning, if either one of these is successful in creating FT, the other one can also get FT and will flower. Okay. So last kind of important thing to know is how do we get the presence or absence of the CO protein? And it has to do with first how CO mRNA concentrations change throughout the day. So if you were to graph CO mRNA throughout the day, it would increase and peak in the late afternoon and then go back down. So it increases throughout the day, peaks, let's say like 4 p.m., and then goes back down. But mRNA isn't the protein, so that doesn't mean the protein's around. In order for the CO protein to be made from this mRNA, you have to get the coincidence of red light. So the key is CO mRNA peak plus red light equals CO protein. So it's this relationship here that creates the CO protein. And when you see red light being important, hopefully by this time you think in your head, oh, I bet phytochrome has something to do with it. And you would be right. Specifically, it's phytochrome in the PFR form, right? That stabilizes the CO protein. So you need high amounts of mRNA and you need the PFR form of phytochrome to stabilize the CO protein. Okay, question?
I had a question going back to that graft situation where the CO protein can travel. So did you say that in the short day plant, it's also going to flower?
Yeah.
Okay. So even that section of the plant is still gonna flower, even though like a full short day plant, CO would typically be a repressor for flowering.
Yes. Because
Okay.
the
Why?
mRNA can travel. So if it's made down here, it can still travel to this part of the plant.
Okay. I was just confused because if it's, I thought CO still being present in the short day plant was gonna just act as a repressor.
It's going to repress making FT, synthesizing the FT protein up here. But there can be enough FT made down here to where it can travel up there. So
so the FT can also travel.
Yes.
Okay, that makes sense. All right, thank you.
You're welcome. So let's go through what an experiment would look like that you might see. You might be given a graph like this. It says CO mRNA. And let's say you're told you have a short day plant but you've grafted on a long day plant. So that little piece is the long day plant. So the short day. Okay, and then you as the experimenter manipulated the light delivered to this plant. So let's say in experiment A, you gave light all through this region and then did some dark here. Then let's say you did another experiment. E, and you did light here and light here. Oops. Okay, and then I would ask you, would you expect flowering in each of these cases? So for experiment A, we have light occurring during the peak of the CO mRNA. So that means that there is the coincidence of peak CO and the PFR form of phytochrome. So because of that, we would expect high amounts of CO in A throughout the whole plant. So in the short day plant, high amounts of CO is going to repress FT. In the little piece of long day plant, high amounts of CO is going to activate FT. So this would cause no flowering in the short day plant, but flowering in the long day plant. But because we made some FT in the long day plant part, that could travel down to the short day plant and we would still get flowering in the short day plant. So if any FT gets made anywhere, then both parts are going to flower. Okay, in experiment B, we had light and then dark, and then a little flash of light right at the peak of the CO mRNA. Because we added this little flash of light, again, we induced the PFR form of phytochrome at the right time. So if you get an increase in PFR at the same time as the peak, then you get increased CO, and it would be the exact same thing. All right, what if I said we did experiment B, but this time the light I used was blue light. How would that change this? Well, if we just shined pure blue light, then there would not be enough red light to convert it to the PFR form. So there is not the coincidence of PFR and peak CO mRNA. So you would get the opposite. You would get lower CO protein in the short day plant, that would de-repress FT, and you would get flowering. Decrease CO in the long day plant would mean less activation of FT. But you would still get flowering because we had some FT made over here. So the long day plant would still flower. Yeah, question?
Yeah, the name of this mechanism is like external oscillator coincidence model. And I had a question about that title. Is the word coincidence referring to the PFR that is made and it coinciding with a Constance protein, or is it referring to the Constance mRNA that oscillates that's coinciding with the light, the red light?
Yeah, it's the oscillating mRNA amounts, and the coincidence is the peak with PFR. Because PFR is what we're saying stabilizes the CO. Yeah, question.
Okay, yeah, just to kind of clarify all of this. So FT is needed for flowering in all plants.
Yes.
Okay, and so in short day plants, a decrease in CO protein increases FT.
Yes.
But in long day plants, an increase in CO protein increases FT.
Yes.
Okay. So in that case, I guess the best adaptation would be for a plant to have both short day and long day plants grafted together, because that way, no matter what, it's gonna get FT, right?
I mean, it just depends on what the goal of the plant would be. Because depending on where it is in the world, it could be advantageous to flower at different times. So we typically
okay.
consider one or the other.
All right. But I guess like in a lab, if we just wanted a plant to flower and you wanted to maximize the chances of flowering, then you would just graft like both types.
Yes, for sure.
All right, yeah, that makes sense.
So they did these grafting experiments usually to investigate whether these molecules involved could travel. And so when they did the grafting and saw these responses, they knew that FT was moving around. Yeah, question.
I just wanted some clarification as to how like PFR and PR relate to the CO. Like how does that affect the CO?
That's a great question, and it's so complicated in real life. But in 220, we're saying the PFR stabilizes the CO protein. So if PFR is around, CO protein made. PR, no stabilization. Anything else besides PFR, no stabilization.
So then to put all together, you would say red light is absorbed by PR and that's converted into PFR. So then the PFR stabilizes the CO.
Yes.
And then it flowers if it's stabilized, or it doesn't flower if it's stable?
Depends on the type. Because in long day plants, the CO protein activates flowering. In short day plants, the CO protein inhibits flowering.
Okay.
Which makes sense, right? Because long plants in light flower. So if we have something like this, a long day of light, that's going to cause the long day plant to flower, or it's going to have the opposite effect on the short day plant, which doesn't flower in long days. The short day plants only flower when there's long nights, because the long nights would cause less CO protein, which means less repression of their flowering genes.
Okay, I think that makes sense. Thank you.
You're welcome. Next question.
Okay, never mind. You actually just answered it right there because I was so confused on the short day plant flowering, but the long, when you mentioned the long nights, leads to less repression. That makes sense. So thank you very much.
Great. Last thing I want to say is for these experiments, yeah, be prepared for different types of light. So normal lighting would be like white light. And you should know normal white light has actually all colors of light, right? Has blue, green, red, far red, but it has more red than far red. So a normal white light, you would expect the PFR to happen. If I give you pure blue light or pure green light or far red light, those will all not work. You need either white light or red light directly to cause the PFR form. Anything else is not going to work. Okay. I feel like I covered everything that you will see come up. If you get everything here, then you'll get 100. Julia, do you have a question or is your hand already raised? Do you have a question?
Oh no,
Yeah.
sorry, I forgot to take it off.
Oh. Cool. Any other questions? All right, well, you guys are gonna do so great and awesome, and I will upload this as soon as I log off. So yeah, feel like I can do great.
Thank you
Thank
very
you.
much.
Thank you.
Of course, yeah.
Thank you.
Real MVP, man. Appreciate you.
Woo. I'm glad to help.